Basic medical sciences, welcome. In this video, we are going to talk about endocrine pancreas. We talked about exocrine pancreas in the previous video, so you can click the link on the top right corner here and watch that video first. Right, so the endocrine part of the pancreas is represented by these green dots. They are actually islets of Langarhans or pancreatic islets. Here you can see a zoomed picture here, right? So peripherally here you see alpha cells. They synthesize a glucagon. Inside you see these green cells. They're actually beta cells. They secrete or they synthesize insulin. Then there are delta cells in yellow. They synthesize somatostatin, and the PP cells for pancreatic polypeptide, and the epsilon cells for ghrelin. Right, so here we're going to have an overview. Right, so the alpha cells, I said they secrete glucagon. Right, what is the function of glucagon? Glucagon has a catabolic effect, right? When is it synthesized? When glucose is gone. Glucagon, when glucose is gone, right? So I said it is a catabolic effect. It opposes the action of insulin, right? So how does it increase glucose? Because I said it's produced when glucose is gone, right? So it increases gluconeogenesis, the synthesis of glucose from non-carbohydrate sources, glycogenolysis, that's breakdown of glycogen, increase lipolysis, and ketogenesis, right? Synthesis of ketone bodies, right? So what stimulates the synthesis of glucagon is hypoglycemia when glucose is gone, and inhibition is opposite, hyperglycemia, and also insulin and somatostatin. These are the inhibitors of glucagon synthesis. Next, we'll talk about the beta cells. The beta cells secrete insulin. Insulin has anabolic effect, is opposite to glucagon, right? So what does it do? It increases the glucose uptake in skeletal muscles and adipose tissue. It increases glycogenesis, right? Synthesis of glycogen, right? So you have a lot of glucose in your blood. You convert it to glycogen, the storage form. It also stimulates lipid synthesis, protein synthesis in the skeletal muscles, potassium uptake into the cells, renal sodium retention, and it decreases lipolysis, right, in the adipose cells. What stimulates production or secretion of insulin is hyperglycemia, when there is high concentration of glucose in your blood. It's also stimulated by beta-2 adrenergic receptor agonism and incretins. What about inhibition of secretion of insulin? This is inhibited by alpha-2 adrenergic receptor agonism, right? Stimulation is beta-2 adrenergic receptor agonism, and inhibition alpha-2 adrenergic receptor agonism, right? So in beta cells, you find both of these receptors, alpha-2 and beta-2. Next, the delta cells. Their function is to produce this hormone called somatostatin, right? So what's the function of somatostatin? It inhibits or decreases the synthesis of insulin and glucagon. It decreases growth hormone secretion, decreases gastric acid and pepsinogen secretion, decreases pancreatic and small bowel fluid secretion, decreases gallbladder contraction, thus decreasing biliary drainage, and it causes splanchnic vasoconstriction. Right, there is a drug which is an analog of somatostatin. It's called octreotide, and this drug is used to treat acromegaly, carcinoid syndrome, and variceal bleeding, right? The patient with esophageal varices, like especially those with portal hypertension, right? So what stimulates secretion of somatostatin is gastric acid, like low pH, right? If it is acidic, and inhibition. All right, so somatostatin is inhibited by vagus nerve, right? So vagus nerve is parasympathetic. It produces acetylcholine, and through a cascade of reactions, it will end up inhibiting the production of somatostatin. Next, we have epsilon cells. These ones secrete ghrelin, and this ghrelin stimulates appetite and hunger, right, in the lateral hypothalamus, and also promotes a release of growth hormone, right, from the anterior pituitary. What stimulates these epsilon cells to produce ghrelin? Fasting, sleep deprivation, right? So if you are awake for a long time, you end up producing this ghrelin hormone, and you will be hungry, you will feel hungry, you will feel the need to eat, right? And it's inhibited by intake of food, right? So this hormone, the production or secretion of this hormone ghrelin, is inhibited by food intake. And the last group are the PP cells, formerly known as the gamma cells, right? So these ones they produce pancreatic polypeptide, and the function of pancreatic polypeptide is increasing gastric secretion of gastric acid, decreasing pancreatic exocrine secretion, and promote satiety, like you feel, you feel full, like as opposite to hungry, right? So it's stimulated by fasting and is inhibited by intake of food, right? So almost the same as ghrelin. Now let's have a closer look on the synthesis of insulin, right? So the insulin gene transcription and translation occur in the rough endoplasmic reticulum in the beta cells of the pancreas, and they will end up producing precursor protein. Signal proteases will remove the signal peptide of proinsulin, then will form proinsulin. Protein folding and formation of one disulfide bridge in the alpha chain and two disulfide bridges between the alpha and beta chains. So this proinsulin will be transported to the Golgi complex or the Golgi apparatus, where it is packed in membrane-bound secretory granules with protein convertases. This proinsulin will be cleaved by proteases, and mature insulin will be released together with C-peptide, right? So this C-peptide is very important. So secretion of insulin and C-peptide is via exocytosis. Right, so look at this pancreatic beta cell. Let's say there is a high concentration of glucose in your blood. So through the GLUT2 transporters, these beta cells will take up this glucose, and through different mechanisms like glycolysis, citric acid cycle, and electron transport chain will form ATP, right? So normally, this sodium potassium pump, right? It pumps like 3 sodium in and 2 potassiums out, right? So in high ATP, we are inhibiting this channel, right? So we're inhibiting this channel which pumps potassium out. It means inside the cell it will be more positive, right? So so you can see this channel is a voltage gated or voltage dependent calcium channel, right? So you can see here is bound, now it's more positive, so the channels are open. The calcium will mediate the fusion of these vesicles with this cell membrane, and through exocytosis we release insulin together with C-peptide. Right, so C-peptide is released by beta cells in an equimolar proportion to to insulin, but is not present in exogenous insulin. It can therefore be used to differentiate between endogenous, thus insulinoma, sulfonylurea use, and exogenous hyperinsulinism, right? So C-peptide is very important. Right, let's talk about the regulation of insulin secretion. The secretion of insulin is chiefly regulated by the levels of serum blood glucose. Insulin secretory pathway, right? So I showed you in the previous slide, right? So through the GLUT2 transporters will take up the glucose into the beta pancreatic cell, which will increase like glucose metabolism, thus increasing ATP and closure of ATP sensitive potassium channels, and the cell membrane depolarization will open the voltage gated calcium channels, which will increase the intracellular calcium levels and increasing insulin secretion, right? So here is what stimulates insulin release and what inhibits insulin release. So the number one stimulant of insulin release is glucose because of incretin secretion following food ingestion. So oral administration will stimulate more insulin release than IV glucose. It's also stimulated by beta agonists, free fatty acids, amino acids, and incretins, for example, GLP-1 and GIP, right? GIP is gastric inhibitory peptide. And what inhibits insulin release? Alpha-2 agonists, epinephrine, cortisol, glucagon, somatostatin, right? So we already talked about alpha-2 agonists, epinephrine and glucagon, somatostatin, and also you need to remember cortisol. Thank you so much. If you like this video, please make sure you hit the thumbs up button and don't forget to subscribe. Peace out.